the previous lecture, we talked about reproduction. In the third lecture, we're going to talk about embryogenesis and development. Starting first, we'll go into the early developmental stages. Fertilization is the joining of sperm and ovum. This creates a functional zygote and will immediately start a cascade of events leading to the formation of a fetus. For the purpose of the MCAT, let's talk about some unique types of cleavages, starting with fraternal twins. This is when two eggs are simultaneously released and fertilized simultaneously. These two eggs will plant into the uterine lining at the same time. Identical twins are when the same zygote splits into two and generates two organisms. These identical twins are going to be genetically identical. For obvious reasons, identical twins are a very popular testing subject when it comes to observing biological systems, especially nature versus nurture studies. Cleavage is the separation of a zygote into a functional organism. It starts off as a morula, which is stated and listed right here, which eventually hollows out and forms a blastula. Lastly, it becomes a gastrula, and that's observed as when the separation cleavage generated creates a pocket. These layers are going to eventually generate different cavities and different organs for the mature organism. As this occurs, it is implanted into the endometrial lining and forms the placenta. And the developing organism is then connected to the placenta by an umbilical cord. Gastrulation leads to the formation of three germ layers, which generate different parts of the organism. The ectoderm becomes the epidermis, hair, nails, and epithelia of the nose, mouth, and anal canal. This also forms the nervous system and the eye lens. A really common way you can remember the ectoderm is calling it the attractoderm, because it's generally things that we find attractive in others. The mesoderm, it becomes the musculoskeletal, circulatory, and excretory system. It also creates gonads and the digestive and respiratory tract tissues. I generally remember this one by remembering it as muscle-oderm. The endoderm is what becomes the epithelial lining for the respiratory and digestive tract. It also becomes part of the endocrine and secretory structures. The endoderm, a shorthand for this one is the inside-oderm, because all these things are inside of the body. Neurulation is the formation of the nervous system. For humans and other vertebrates, it involves the formation of the spinal cord. Now that we've covered some early developmental stages, let's talk about mechanisms of these developments. Cell specialization follows very specific concerted pathways. Determination is when the cell commits to a specific cell lineage. And differentiation is when the cell undergoes changes due to a selective transcription to take on these appropriate characteristics. Note that differentiation occurs after determination. Stem cells are classified into three different types. There's totipotent, pluripotent, and multipotent, and the definitions are listed below. When it comes to intracellular communication, there are different types, and these communications are going to suggest and help with things such as differentiation. The autocrine communication pathway is when the signal acts in the same cell that released the signal. Paracrine is when a signal acts on adjacent cells. Juxtacrine, acting through direct stimulation, and endocrine is when it acts on distant cells by going through the bloodstream. A really common form of endocrine communication is, as you would guess, hormones. Lastly, apoptosis. A cell that's generating this sort of signal is going to program its own death. This is generally if the cell deems that it's beyond a point of repair, or if other cells tag it as something that is incorrect. One thing we can talk about when it comes to this sort of differentiation and division and development is cancer. Cancer is when this differentiation or replication is uncontrolled. If there is a carcinogenic cell, it'll often trigger some form of apoptotic mechanism. When we're talking about the fetus, it's imperative that we talk about its circulation. This is all undergone through the placenta, and because of the concentration gradients, the substances are passed accordingly. Fetal hemoglobin, in particular, is a common testing subject. It has a higher oxygen affinity than adult hemoglobin, which explains why maternal hemoglobin can successfully pass oxygen to the fetus. Be aware, though, that the arteries and veins of the placenta, the umbilical arteries and umbilical veins, are reversed from what they are commonly associated with in the adult's body. Arteries generally carry oxygenated blood in the human body, and veins deoxygenated. However, when it comes to umbilical arteries and umbilical veins, these roles are reversed. Note that arteries carry blood away from the heart, and veins carry blood to the heart. For the intents of a fetus, the veins are what carries oxygenated blood to the heart, and the arteries carry deoxygenated blood away. This doesn't reverse until they are actually born. The last portion we'll go into in this lecture is gestation and birth. Gestation is, as you would suggest, is the cultivation and growth of the fetus. It's divided into trimesters in humans, and there's 3 because a pregnancy lasts about 9 months. The first trimester is when organogenesis occurs. This is when organs are slowly starting to be created. Soon afterwards, you'll start to see the beginnings of eyes, gonads, limbs, and liver. Roughly, the infant by 5 weeks is going to be about 10 millimeters, and by week 6 it'll be about 15. A skeleton does start to form here, but it's cartilaginous. The second trimester is when growth happens exponentially, and the fetus does start to move, and a face starts to become distinctly human. At the end of this 6 months, you'll generally see about a 30 to 36 centimeter long fetus. The final 3 months, the last trimester, involves another rapid growth cycle and brain development. This is also when antibodies are transferred to the fetus from the mother. The last thing we'll go into is birth. Birth for humans is done via labor, which is when the fetus is pushed through the vagina along with the placenta and the umbilical cord. This is triggered by contractions in the cervix, which causes it to expand. When the infant is ready, the amniotic fluid in the sac is going to burst, and the infant is going to be pushed out through the vagina. There are obvious complications with this, which is why processes like a C-section are required at times. So that'll cover embryogenesis and development. There's a lot of things that we sort of glossed over, so it might be a good idea to go back to your biology lecture textbooks and just take a good look over some of these processes. It'll definitely benefit you on test day. Please join us in the next lectures as we go into some of these systems in the human body.